All right, for the past 2 summers, I've worked over at the University of Nebraska Medical Center in a cardiovascular physiology lab. So my research basically focuses on the cardiovascular system and how its function is controlled by the central nervous system. There are a few technical words up there in that title, but I'll do my best to explain them to you in layman's terms in the next 10 minutes. So simply put, the area that we're interested in is heart failure, which is the inability of the heart to pump enough blood to meet your body's needs. Over time, conditions such as narrow arteries in the heart, which is called coronary artery disease, or hypertension, can leave your heart unable to pump enough blood to meet your body's needs. It's a leading cause of death, affects nearly 5 million Americans. It's also a very costly disease to the American healthcare system, upwards of $35 billion annually. The body usually regulates cardiac function actively, using both hormones and signals from the nervous system, which act to maintain equilibrium or homeostasis. So for the non-science people out there, think about your thermostat. If it gets too hot in your house, your thermostat senses it and acts to correct things. And that's basically what we do in physiology. We think a lot about negative feedback loops. So my particular area of interest is the Baroreflex, which is one of the body's homeostatic mechanisms for controlling blood pressure. It provides a negative feedback loop, much like the thermostat on that last page, in which elevated blood pressure causes heart rate and blood pressure to decrease. And then sympathetic nerve activity provides the language that allows those changes to interact. This function is impaired in heart failure by central activation of the renin-angiotensin system, leading to increased production of the peptide angiotensin II. So the question that I was looking at this past summer was whether another peptide, angiotensin 1,7, can help in heart failure. And angiotensin 1,7 is a derivative of angiotensin II, which my lab has previously shown can enhance the baroreflex control of heart rate. So my particular project this summer was looking at how sympathetic nerve activity is specifically indicated in these changes. Here's the overall schematic for the regulation of the Baroreflex. A lot of complicated diagram up there, but basically think of it as a seesaw. We have a balance between two counteracting parts of the cardiovascular system. Angiotensin II on the one hand, angiotensin 1,7 on the other. And both in the heart failure and normal states, both peptides are present. It's about getting the right balance so that your heart's working hard enough without wearing out. So my hypothesis this summer was that central administration of the peptide Ang1,7 would restore the proper balance by decreasing renal sympathetic nerve activity and enhancing the baroreflex control of nerve activity in heart failure. So to confirm this hypothesis, I used a chronic animal model, which were New Zealand white rabbits. Those bugs will work for presentation. So we implanted the rabbits with brain cannulae, which allowed for central drug infusion, pacemakers, which allowed us to induce heart rate in a certain class of the rabbits, renal nerve electrodes, which allowed measurement of renal nerve activity, and arterial pressure transmitters, which allowed us to measure both the blood pressure and the heart rate. So I used 4 experimental groups, first based upon which my drug was, either angiotensin 1,7 or a control of cerebral spinal fluid, and whether I would pace the rabbit and therefore induce heart failure or perform a sham operation. 2 to 4 weeks after these implantation surgeries, I would carry out a baroreflex experiment where I would track the changes in renal sympathetic nerve activity, blood pressure, and heart rate. And basically what that experiment entails is a chemical stress test. So I would give intravenous infusions of 2 different chemicals, sodium nitroprusside and phenylephrine. And they would directly change the rabbit's heart rate. And the heart rate, as the heart rate changed, both the renal sympathetic nerve activity and the blood pressure would have an opposite change. So it's an inverse relationship. So as the blood pressure would rise due to the infusion of phenylephrine, the heart rate would drop and renal sympathetic nerve activity would decrease. S&P would have the opposite effect. So the relationship between these 3 factors I could graph, and that would allow me to compare the responses between different animals. because all the animals would have the same responses. So what I'm looking at is the differences in the responses, the rate at which things are changing. So this would be what I would be saying during one of the baroreflex experiments. You can see the drug is driving the blood pressure to increase here, so in this case it's SNP. But at the same time, you can see the inverse relationship with the heart rate and then different metrics measuring renal sympathetic nerve activity. So from our results, we would plot them on a graph like this, where we look at the changes in heart rate versus the changes in blood pressure. So the first result that we found was that, as expected, baroreflex control of the heart rate is enhanced by central infusion of angiotensin. You can see how the angiotensin lines, those are the red lines, have an enhanced response, whereas the control was more limited. Now this occurred in both heart failure and sham rabbits, which was somewhat unexpected, because in early heart failure, there's an increased sympathetic tone. The second thing we looked at was baseline activity of the renal nerve outside of the experiment. So this is just as expressed as percentage of maximum response. And we found that angiotensin 1.7 reduced the baseline sympathetic tone. This decreased activity could also explain the enhanced baroreflex control that we've seen in the first chart. Our final result was that angiotensin depressed the baroreflex control of nerve activity in both classes. You can see how the drug rabbits have much wider curves. This is consistent with the antagonist role of angiotensin 1.7 versus angiotensin 2, and the idea of balance that we talked about earlier. So even though what I found out this summer is just a tiny little shift in a physiological curve, there are larger implications. In early heart failure, the body ramps up renal sympathetic nerve activity to compensate for lower cardiac output. So because your heart's not working as well, your body will try to ramp up sympathetic nerve activity so we get more blood going through the system, which helps at first. The problem is it wears your heart out because it's not used to that type of stress. So the hope is that administration of drugs such as angiotensin 1.7 in early heart failure may help prolong the lives of people suffering from heart disease. The caveat with these studies is, since it was only 10 weeks of work, I only had an N of 2 to 3 for each of my classes, so I cannot yet talk to statistical significance. So I spent my summer.
Dan, would you take any questions?
How easy is it to move from this animal model to a human model?
It's actually not as hard as you'd expect. Currently, the lab that I was working in has a collaboration with the lab at the University of Missouri, and they're actually doing human trials right now.
How many subjects did you have in your groups?
2 to 3.
That was the end that you just mentioned?
Yep. So about, I think, 12 rabbits overall is what we had.
Yeah, okay.
And would you describe, like, you know, the experimental procedure? How much time did it take you to prep? What were you on? And then when was your day over, so to speak, in that experiment?
Well, the first thing we'd have to do is have a scheduled surgery to implant the different physiological tracking tools that we'd need. And, you know, to prep the rabbit would take probably an hour, and the surgeries would take 3 or 4 hours. And then we'd have to wait to allow the rabbit to recover so the effects of anesthesia would wear off. And we'd give them 2 or 4 weeks to recover. So it took a lot of planning to figure out when we were going to schedule surgeries and then when we were going to schedule experiments.
Did you perform the surgeries?
I assisted. So I would open and I'd close and I'd poke things around, but I also had a doctor helping me.
Do you anticipate that this will have any role to play in life after Creighton?
You know, I learned how to suture and I learned how to intubate rabbits, so it's not something a lot of people can say.
What happened to the rabbits afterwards?
They would be euthanized. And funnily enough, they're not from New Zealand. I don't know why they called that.
I love your cartoon. Thanks, Greg.